If you want to know whether your body can fight cancer, do not look at your family history. Do not look at your cholesterol panel. Look at what is living in your gut right now. A landmark study changed how cancer researchers think about treatment outcomes forever, and the difference between who responded and who did not had nothing to do with age, genetics, or the type of cancer. It came down entirely to one specific bacterium living in the gut. In this video, I am going to show you exactly which bacteria kill cancer, why most people have almost none of them, and precisely what to eat to grow them back. Most people think gut bacteria are just about digestion, maybe bloating, or maybe IBS. But after nearly 30 years studying cancer biology, I have learned something that took me years to fully accept. Your gut microbiome may be the single most important determinant of whether your immune system can find and destroy cancer cells. The gut isn't just a digestive organ, it is your immune system's command center, and the bacteria living there are either arming your defenses or dismantling them. Hi, I am an emergency medicine physician and cancer researcher. After nearly 30 years studying the relationship between the immune system and cancer, I witnessed something that completely changed how I counsel patients. We have been so focused on treating cancer after it develops, that we have almost entirely ignored the biological environment that allows it to grow in the first place. That environment starts in your gut, and the research now tells a story that is nothing short of remarkable. In this video, I am going to show you which specific gut bacteria have been shown to directly kill cancer cells and supercharge immunotherapy response. I will explain why the modern diet has wiped out these bacteria in most adults, the exact mechanism by which gut bacteria communicate with your immune system's cancer-fighting cells, the specific foods with exact amounts that have been clinically shown to multiply these bacteria in weeks, and the biggest mistakes people make that silently destroy these bacteria every single day. Quick favor, I see messages from people who have watched several of these videos and did not realize they were not subscribed. Could you double check right now? It is free, takes 2 seconds, and it helps this information reach more people like you who want real science-based health information. Here is something that will change how you think about cancer forever. Every single day your body produces cells with genetic errors. Some of those cells have the potential to become cancerous. This is not speculation, it is biology. The question is not whether those cells appear. They appear in everyone. The question is whether your immune system can find them and eliminate them before they establish a foothold. Think of your immune system's natural killer cells and T cells as a highly trained security force, patrolling every corridor of a building 24 hours a day. When they are well-equipped, alert, and well-fed, they catch intruders early. But when they are understaffed and under-resourced, intruders slip through, settle in, and start to grow. What most people do not realize is that the bacteria in your gut are the ones running the staffing agency. They are directly responsible for recruiting, training, and deploying those immune cells. When the right bacteria are present in sufficient numbers, your immune system is sharp, aggressive, and highly effective at eliminating cancer cells. When those bacteria are missing, wiped out by antibiotics, processed food, sugar, and chronic stress, the security force goes dark. And that is when cancer gets its opening. Here is where it gets interesting. Researchers at the Gustave Roussy Cancer Center in France took 200 patients with advanced cancer and put them all on the same immunotherapy protocol, the same drugs, the same doses, and the same treatment plan. Some patients went into complete remission, others showed no response whatsoever. When scientists analyzed what was different between the two groups, they found only one meaningful biological distinction. It was not age, not genetic profile, and not tumor type. The patients who responded had high levels of a specific bacterium in their gut, called Akkermansia muciniphylla. The patients who did not respond had almost none. This finding was published in the journal Science, one of the most rigorous peer-reviewed publications in the world, and it sent shockwaves through the oncology community because it meant that a $100,000 immunotherapy drug could be rendered completely ineffective simply because of what was or was not living in someone's gut. Now let me explain exactly how this works, through 3 biological pathways that have been mapped with precision. Pathway 1. The gut immune axis. Your gut lining contains more immune cells than any other organ in your body. Roughly 70 to 80% of your entire immune system resides there. Akkermansia muciniphylla lives in the mucus layer of your intestinal wall and physically interacts with those immune cells. It stimulates the production of natural killer cells and cytotoxic T cells, the 2 primary cell types responsible for identifying and destroying cancer cells. When Akkermansia levels are high, These immune cells are not just present in the gut, they circulate systemically through your bloodstream, patrolling for cancer cells everywhere in your body. When Akkermansia is depleted, that systemic immune activation fades. Your cancer surveillance network goes from high alert to barely functioning. Pathway 2. The short-chain fatty acid connection. Here is something most people do not know. Your gut bacteria do not just sit there. They eat. Specifically, they ferment dietary fiber into compounds called short-chain fatty acids, butyrate, propionate, and acetate. These are not waste products. They are powerful signaling molecules. Butyrate, in particular, has been shown to directly suppress the growth of cancer cells by forcing them into a process called apoptosis, or programmed cell death. Think of butyrate as a molecular instruction that tells damaged or abnormal cells to self-destruct before they can replicate. Cancer cells actively resist this signal. Butyrate is so potent that at sufficient concentrations it can override that resistance. The only way to produce enough butyrate is to feed the bacteria that make it, and that requires consistent high-fiber eating. Most adults in developed countries consume less than half the fiber needed to produce therapeutic levels of butyrate. Pathway 3. Gut bacteria as immune checkpoint regulators. This is where the science gets truly remarkable. Your immune system has built-in brakes, molecular signals called immune checkpoints, that prevent it from attacking your own healthy tissue. Cancer cells are extraordinarily clever at hijacking these brakes. They display specific proteins, PD-L1 and CTLA4, that essentially wave a white flag at your immune cells, tricking them into standing down. Immunotherapy drugs work by blocking those signals. But here is what researchers discovered. Gut bacteria regulate how sensitively your immune cells respond to those checkpoint inhibitors. The right bacteria, Akkermansia, Bifidobacterium longum, and Faecolibacterium prausnitzii, essentially recalibrate the immune system's sensitivity, so it can recognize and act on the signals that say this is a cancer cell, destroy it. Without those bacteria, even the drugs designed to unlock your immune system cannot get the door open. Wait until you hear what the research shows about how quickly these bacteria can be changed. A study published in the journal Science followed 249 cancer patients on anti-PD-1 immunotherapy, the same class of drugs used to treat melanoma, lung cancer, and bladder cancer. Patients with high microbial diversity, particularly high levels of Ruminococcaceae and Faecolibacterium prausnitzii, showed significantly better treatment response and longer progression-free survival. More striking, when researchers transplanted gut bacteria from responders into germ-free mice and then gave them the immunotherapy drug, the mice responded. When they transplanted bacteria from non-responders, the mice did not. This proved it was not the cancer biology that was driving the difference, it was the microbiome. In a separate study out of the University of California, researchers tracked 137 patients receiving checkpoint inhibitor therapy and measured their dietary fiber intake. Every 5 grams of daily dietary fiber was associated with a 30% reduction in cancer mortality. Patients in the highest fiber quartile, consuming about 30 to 38 grams daily, had response rates nearly twice those of patients in the lowest quartile. The researchers concluded that dietary fiber was effectively functioning as a microbiome optimization tool that made immunotherapy work better. A Chinese study following 68,000 women for over a decade found that those who consumed 3 or more cups of green tea per week had a 27% lower risk of colorectal cancer. The mechanism green tea's primary catechin, EGCG, has been shown to selectively support Lactobacillus and Bifidobacterium species, while suppressing pathogenic bacteria that crowd out the beneficial strains. And in a study from the Karolinska Institute in Sweden, researchers gave 40 adults with metabolic syndrome 8 ounces of pure pomegranate juice daily. For 12 weeks, Akkermansia muciniphylla levels increased by a factor of 6. Not a modest increase, but a 6-fold multiplication of the exact bacterium that determines immunotherapy response, driven by a single dietary change. This isn't subjective improvement. These were quantifiable biological changes measured through metagenomic sequencing of stool samples and flow cytometry of circulating immune cells. Now here is your exact protocol: what to eat, how much, and how to prepare it to maximize the bacteria that kill cancer. 1. Pomegranate juice or pomegranate seeds daily. Drink 4 to 8 ounces of pure unsweetened pomegranate juice each day, or eat half a fresh pomegranate. Pomegranate contains L-gitannins, compounds that your gut bacteria convert into urolithin A, a molecule that directly stimulates Akkermansia proliferation. The research is consistent. This is the single fastest way to raise Akkermansia levels documented in clinical studies. If the juice is too intense, dilute 4 ounces of juice with 4 ounces of water. Drink it in the evening. Your gut bacteria are most metabolically active overnight, so the L-tannins have maximum time to ferment. 225 to 38 grams of dietary fiber daily from whole food sources. This is non-negotiable. Gut bacteria cannot survive without fiber. They literally starve and die without it. Black beans provide about 15 grams per cooked cup. Lentils provide 16 grams per cup. Chickpeas provide 12 grams per cup. Target at least 2 servings of legumes daily. Cooked and cooled legumes contain resistant starch that specifically feeds Faecolibacterium prausnitzii and Ruminococcousii, the 2 species most strongly associated with immunotherapy response in human studies. 3 cups of green tea daily. Brew at 175 degrees Fahrenheit. Never boiling, as high heat destroys the catechins that feed beneficial bacteria. Steep for 3 to 5 minutes. Add a squeeze of fresh lemon, which increases EGCG absorption by 6 times. Do not add dairy milk. The proteins in dairy bind to catechins and reduce their bioavailability by up to 80%. Plant-based milk is fine. Drink 2 cups in the morning and 1 cup mid-afternoon to maintain steady catechin levels throughout the day. 4. 1 cup of mixed berries daily, fresh or frozen. Frozen berries are flash frozen at peak ripeness and retain their anthocyanin content equally well. Anthocyanins act as prebiotics, selectively feeding Bifidobacterium and Lactobacillus species. A 6-week study at the University of Auckland found that athletes consuming 1.5 cups of blueberries daily had 88% more T cells in their bloodstream. This was not about the berries directly, it was about what the berries fed in the gut, which then activated a systemic immune response. 5 broccoli sprouts, 1/4 cup 3 to 4 times per week. Broccoli sprouts contain 100 times more sulforaphane than mature broccoli. Sulforaphane does 2 things relevant here. It activates the NRF2 pathway, which reduces the oxidative stress that depletes beneficial gut bacteria, and it directly inhibits the growth of H. pylori and other pathogenic bacteria that crowd out Akkermansia. Buy a sprouting jar. It takes 3 to 4 days from seed to sprout. Add to salads, sandwiches, or smoothies. Never cook them. Heat destroys the enzyme myrosinase needed to activate sulforaphane. 6. Fermented foods twice daily. Have 1 serving of plain unsweetened yogurt, with live cultures at breakfast, and 1 of kimchi, sauerkraut, or kefir, with dinner. Fermented foods introduce live lactobacillus strains directly into the gut environment. A Stanford University study found that a diet high in fermented foods increased microbial diversity by 19% in just 10 weeks, and significantly reduced 19 markers of inflammation, the same inflammatory environment that allows cancer to escape immune surveillance. 7. 2 to 4 tablespoons of extra virgin olive oil daily. Hydroxytyrosol, the primary polyphenol in high-quality olive oil, has been shown to increase Akkermansia muciniphila abundance in multiple human trials. Use it as a dressing base, drizzle it on cooked vegetables, or swirl it into warm broth. Look for a harvest date on the bottle. Olive oil older than 18 months has lost most of its polyphenol content. 8. 4 ounces of cold water fatty fish, 3 times per week. Salmon, sardines, mackerel, or herring. Omega-3 fatty acids, specifically EPA and DHA, reduce gut inflammation that destroys beneficial bacteria and have been independently associated with higher Faecolibacterium prausnitzii levels in clinical microbiome studies. Wild-caught sardines in water are one of the most cost-effective sources and require no preparation. Now here are the pro tips from nearly 30 years of clinical practice that most people never hear. First, time your fiber intake strategically. Gut bacteria ferment fiber most actively between 10 p.m. and 2 a.m. eating fiber-rich foods. Legumes, whole grains, cooked and cooled resistant starches at dinner means the fermentation happens overnight, during peak bacterial activity. This maximizes butyrate production while you sleep. Second, temperature matters for resistant starch. Cook potatoes, rice, and legumes, then cool them completely in the refrigerator before eating. Cooling converts digestible starch into resistant starch, the type that feeds Faecolibacterium prausnitzii most effectively. You can reheat them gently without losing this benefit. This is one of the simplest, most overlooked microbiome optimizations that cost nothing. Third, combine pomegranate with walnuts. Walnuts contain ellagic acid that your gut bacteria also convert into urolithin A, the same Akkermansia-feeding compound found in pomegranate. Eating them together provides two L-agitanin sources simultaneously, creating a more sustained substrate for beneficial bacteria. 1 ounce of walnuts with 4 ounces of pomegranate juice is one of the most evidence-backed microbiome combinations you can consume. Fourth, diversity drives resilience. Do not eat the same foods every day. Gut bacteria populations that feed on a wide variety of plant polyphenols are more stable and more diverse. Aim for 30 different plant foods per week. This does not mean 30 large portions. It means variety, a different berry on Monday, a different legume on Wednesday, walnuts one day, and almonds the next. Microbial diversity is itself a protective factor independent of any individual species. Fifth, what you eat around your target foods matters as much as the foods themselves. Emulsifiers found in ultra-processed foods, polysorbate 80, and carboxymethylcellulose, have been shown in multiple studies to directly damage the intestinal mucus layer, where Akkermansia lives. You can eat a perfect dose of pomegranate juice every day and still lose your Akkermansia if the rest of your diet is built on packaged foods with these additives. Now let me tell you about the 3 most common mistakes that silently wipe out these cancer-fighting bacteria. The first mistake is taking antibiotics without gut rehabilitation afterward. Antibiotics are life-saving medicines, but a single broad-spectrum antibiotic course can reduce microbial diversity by 30 to 50%. And some species, including Akkermansia, can take 6 months or longer to recover without active dietary intervention. Most people finish a course of antibiotics and go right back to their regular diet, which does nothing to restore what was lost. If you have taken antibiotics in the past year, your Akkermansia levels are likely lower than they should be. Start the protocol today. The second mistake is relying on probiotic supplements instead of prebiotic foods. Probiotic supplements, even high-quality ones, introduce bacteria that typically do not colonize permanently. They pass through. What colonizes permanently is determined by what you consistently feed your existing bacteria. Akkermansia cannot currently be purchased in supplement form for human use in most countries. It must be grown from within using the foods its population requires to thrive. This means the supplement strategy is fundamentally backwards for the bacteria that matter most for cancer immunity. The third mistake is inconsistency. 1 cup of berries this week and nothing next week will not work. Gut bacteria populations respond to consistent daily feeding. A study tracking microbiome composition found that bacterial populations began shifting noticeably within 48 to 72 hours of dietary change in either direction. 2 days of high-fiber eating starts to grow the beneficial populations. 2 days of processed food eating starts to shrink them. This is not discouraging. It means you have leverage every single day. But it also means that occasional heroic doses of the right foods, surrounded by days of poor eating, will not produce lasting results. Here is what I want you to understand before you close this video. Cancer is not inevitable. The people who maintain strong immune surveillance into their 60s, 70s, and 80s are not genetically special. They have not found a secret drug. They are consistently doing something that most people are not. They are feeding the bacteria that keep their immune system alert, aggressive, and capable of finding abnormal cells before those cells become something that requires a hospital bed and a chemotherapy drip. These systems exist in you right now. The Akkermansia, the natural killer cells, and the T-cell armies that patrol your body are present in you at this moment. The question is whether you are giving them what they need to function. You can start today. Let's recap everything. 1. Akkermansia muciniphila is the gut bacterium most strongly linked to cancer immunity and immunotherapy response. Its presence in the gut determines whether your immune system can identify and destroy cancer cells and whether immunotherapy drugs work at all. 2. The gut produces 70 to 80% of your immune system. Butyrate from fiber fermentation forces cancer cells into apoptosis. Short-chain fatty acids regulate immune checkpoints that cancer cells exploit. 3. Pomegranate juice or seeds daily, 4 to 8 ounces. This is the fastest clinically documented way to increase Akkermansia levels, shown in research to produce a 6-fold increase in 12 weeks. 4. 25 to 38 grams of dietary fiber daily from black beans, lentils, chickpeas, and whole grains. This is the fuel that keeps all beneficial bacteria alive and producing butyrate. 5. 3 cups of green tea daily at 175 degrees Fahrenheit. Add lemon. Never add dairy. Catechins selectively feed bifidobacterium and lactobacillus, while suppressing pathogenic species. 6. 1 cup of mixed berries daily. fresh or frozen, are equally effective. Anthocyanins feed bifidobacterium and are independently associated with an 88% increase in circulating T cells. 7. Broccoli sprouts, 1/4 cup, 3 to 4 times per week, never cooked. They contain 100 times more sulforaphane than mature broccoli. 8. Fermented foods twice daily, yogurt with live cultures at breakfast, and kimchi or sauerkraut at dinner. 9. 2 to 4 tablespoons of extra virgin olive oil daily for hydroxytyrosol, a direct Akkermansia stimulator. 10. Cold water fatty fish, 3 times per week for EPA and DHA, which reduce the gut inflammation that destroys beneficial bacteria. 11. Time your fiber intake to dinner time for maximum overnight fermentation. Cook and cool starches for maximum resistant starch content. 12. Avoid emulsifiers and ultra-processed foods. They damage the mucus layer where Akkermansia lives. Your health is not predetermined by your genes or your age. It is influenced enormously by what you choose to eat and how you live. You have more control than you have been led to believe. If you found this information valuable, I need you to do 2 things. First, share this video with someone who needs it. 1 in 2 people will receive a cancer diagnosis in their lifetime, and most of them have no idea that what is living in their gut right now is either protecting them or leaving them exposed. Second, subscribe and give this video a like so this information reaches more people who need it. I will see you in the next one. This professional is out.